Hello, good afternoon, everyone. Welcome to Liquid Brain. So today is going to be a part 2 of Mother 16S rRNA analysis, particularly on taxonomy and how to actually do metagenomic analysis once you have done the sequencing. So this is going to be a part 2 on analysis and visualization of the data. So if you have not watched part 1, make sure you understand how to do data cleaning and preparation. Otherwise, you might want to finish that part before you come over. So where we actually stopped is that once we finish up cleaning all the data from the fast queue, we align them, we check for error rates and all that thing, we end up with these 3 files in our directory. So we have a final FASTA, which contain all the sequence of all the contigs that we have done assembly. We have a final count table, which actually show that how many times each of the contigs has appeared in all of our sample. And we have a final taxonomy, which actually tells you the taxonomy information, like the kingdom, phylum, genus, and all that stuff in our FASTA file, which all the different contexts, where do they belong to and how they are doing. So the analysis is going to be something on like this 7 step. They're not actually equal, but the flow will be somewhat like that. So half of it, 1, 2, 3, 4, is going to base on the MySec SOP, while 5, 6, 7 is going to base on R and Galaxy and some of the website that I found. So the first thing we're going to do is to look through the file that we have developed from our last analysis to make sure they're in the correct format. And if it's not the same as what you have, you might want to look into your analysis pipeline or try to do some data manipulation to make sure you end up with the same format that I have. So we're going to do some OTU classification in Mother. We're going to do a phylotype and phylogenetic studies, particularly in the alpha and beta diversity of different sample. And then we're going to move forward to RStudio using plotting trees in RStudio using like ggtree. There's a phylogenetic tree. We're going to do a little bit of graphing Galaxy using Krona, and we're going to visualize most of the data in Megan based on the output that we have using using Galaxy to actually do a microbiome, because that's, I believe, some version issue. So the main goal for you watching this video is not just to generate those files, but rather to understand the format and output, why they're this way, and how do you convert them from one way to another. Understanding the purpose of the analysis pipeline. Why are we running refraction? Why the beta and alpha diversity? What do that stands for, and why do we do those things? And the next one is analysis and visualization of taxonomy information between sample. How does one sample differ from another? How do you make different trees? How do you do a coloring, clustering, clades, and all that stuff? And the last will be a visualization of result, particularly the Krona and the phylogenetic genetic diversity, the diversity of taxonomy within our sample. So some of the output that we expect is to actually load all this file into our R, or at least open up in text file and understand what are the contexts, the count per sample. That's our count table. What is the context of our taxonomy? What's the distance between the contexts that we have? We do an OTU count and taxonomy table. We're going to import all this into R properly with proper tools, and then we're going to generate a simple heat map just to show you how to manipulate the data. We're also going to plot some trees in ggtree, something like that, and do a little bit of customization on the size and all that stuff so that it's easier for you to present your data later in journal. And we're going to move on to Galaxy, which is a tool called Krona, so that it can actually show you all the diversity and taxonomy within the sample. They're actually a group of all the samples that you have, so not one by one. And lastly, we're going to do a make biome. We're going to export a biome file. We're going to use another pre-existing tool called Megan, which is free. So you can actually make all the beautiful graph that you see right here. So that's basically introduction to all the thing. I believe there will be timestamp in the video description down below or just on the timeline. So do skip on to the step that you want. Otherwise, I'm going to move on to Mother, and we're going to start our analysis on looking at our output. So if you recall, last time we actually stopped at this step over here in the MySec SOP, which is rename the current FASTA, count, and taxonomy into something called a final dot whatever, so that we can actually proceed with the analysis. So final means that it's finally ready for downstream analysis because it has been cleaned enough. Okay, so you should have three files over here: final taxonomy, final FASTA, and final count table. So since our data here is relatively small, you can open it directly in the text pad. But in big file, I'm gonna show you how to open in R later. So let's actually look at first our FASTA file. So FASTA file will have first of all the name or the context that has been assembled, as well as all the different sequences underneath it. Okay, so I think there's about 12,000 different of them. So if you scroll through them, it's going to take a long time. And usually we don't actually look at raw data like that. They're more of like that automated tool for you to do that. Second is the taxonomy file. You can actually see that these are again the same name that we have seen in the FASTA file just now, but instead of sequences, we get to know what is their taxonomy, what is their kingdom, phylum, class, and so on and so forth. So we also have the count table, which actually means that how many times does each of the sample appear in each our sample, in each how many times the context has appeared in each of our sample. And you can see these are the name of the sample, and 119 means that this context appear in 19 and 1 times in each of them. So I'm going to show you how to actually create a sparse matrix later, so that's easier to understand what do they do. And instead of actually looking at them like that, you can also go to R and actually try to import them as a delimited CSV or something like that. So what you need is actually two library over here, tidyr and readr. So for those that don't want to write their own, you can actually download this script again from my GitHub page in the video description down below. So you can just do a read delimited on our count table and taxonomy just now, and you can very easily get something like this, where you have the context and you have the kingdom, phylum, class, and order. So if you recall just now, because there are actually just two different columns when you import them, you can actually run a separate command over here to actually separate column 2 based on the semicolon into this separate different column, and you can actually have a better look at the every context as well as their phylum. So this is particularly useful when your context file or count file is extremely big, you're unable to open them in your notepad. Okay, so you can run the same thing with the context as well as the count file, and I have done a very simple heat map over here. So you actually can have a look at the heat map between all the sample and how many times each of the context appear in those sample and how they're clustered, and you can have a basic idea on how every sample are actually different or similar to each other based on this table alone. Okay, so that's the that's the three file format that we're gonna start with, which is the FASTA, the count right over here, and we also have the context the context right over here and the count right over here. So this is actually what we'll be starting to work with. Okay, so let's actually move on to a first step of our analysis, which is the OTU, which is the Operational Taxonomy Unit or something like that. I'm not too sure. But basically what they do is they try to find conserved sequence in between our FASTA file and actually try to assign some region between them so that you can actually use those region to see how similar are they for each of them. So they are actually called OTU, which is a section of those sequences. So the idea is pretty simple. We're gonna run a distance sec to actually get the distance between them. This is going to take quite a while, so I'm not going to run it in real time here, but you will get something like a final dot this by the something like that, final dot this when you're finished running it. So I'm going to run the second cluster function here directly so that it's going to be a little bit faster. So what these two trying to do is to find the distance between each of our each of our context, and actually cluster them to find the conserved sequence, the conserved region and OTU that is within our sample. Okay, so cluster split is the same as above, so you can actually skip that. But the next one is actually do a make share. So make share is very important because make share is the step where actually makes the file format easier to be export or share among different program, and it's also something that is easier for human to read. And once we're done the make share, we have a share file, final optmcc.share. Just remember, it's going to be very useful later on. And once we've done that, we're going to do a classified OTU over there to actually finally get our OTU table out here. Okay, so once you run, you also have a cons taxonomy, also very important. We're gonna open them and have a look right here. Okay, so what does this table means is that you have different OTU, which is like a section of the gene. You have the size of this OTU and the exact taxonomy of what they are, and actually is being arranged in the decreasing order. So you have OTU 1 to OTU maybe 102 or something like that, and how many times it appear and what exactly are there. So it's again very difficult to actually see data like that. So I've written a simple R code right here as well to actually read the OTU into our program, and you can have a look. It's exactly the same thing, but the difference now is that I'll be able to just run a script to actually have a look on what kind of class or kind of different bacteria or different kingdom that exist in my data set. And you can see that most of them on a higher count belongs to Bacteroideae, well, and a lot down in the middle are actually called Stridia, and you have something in and out from in between. So you can have already a basic idea of all the OTU and which taxonomy do they actually belongs to. Okay, so you also have one T7 class in Certia and Satis. I'm not too sure what that means. Yeah, it's also time to make a disclaimer that I'm not actually a metagenomic cis. I mostly deal with R programming and like human data and stuff like that. Okay, so once we're done that, we get our classified OTU. We'll have our cons taxonomy table, as we can see from the file output as well as just now. And we also have something called the mcc.share. So these two are very important for a lot of downstream analysis, but now we're going to switch on to a little bit onto the alpha and beta diversity and the phylogeny and phylogenetics based on the MySec SOP. Okay, so this will be our next step. We're gonna completely skip over the ASV because they're basically very similar to the concept of OTU, but certain analysts actually use them slightly differently. So I'm gonna skip over them and we're gonna focus on the OTU analysis. So what the next one is phylotype. So phylotype, I don't have a good way to explain that, except they're just a different way of classifying sequences, though they group them into different group. You can have a look at final.tx.list over here. Here. So you can actually have a look at these are the different phylotype and what are the sequence that contains for each of the sample. So that's basically the same. It's just basically very similar to not very similar to OTU. Yeah, it's very similar to the classification of OTU, just based on a different algorithm on how they're being clustered. Okay, so again, you can actually you can again use the, what is that called? Classified OTU again to figure out the OTU distribution in each of the phylotype that we have. Okay, so it will be in different taxonomy, and if you open those taxonomy, it's going to actually be, for example, it's going to be exactly the same as our OTU table as we've seen just now, just that all this belongs to the third phyllo group in this case. So if you know what you're doing for, you can go for the phylotypes, but I'm not going to talk too much about that. So the next step is actually talk about the phylogenetics. There's some problem with these two analysis where I can't actually run it properly. So I'm going to skip over, but what is this is supposed to generate a database or a sequence data so that you'll be able to plot a phylogenetic tree on an individual sequence that you have. But perhaps the number is too big or just my computer is just not good enough. But please run this on your computer, and if you're able to run the output, maybe just leave a comment down below or just link a video to what you have done, because I can't get a clear cut to finished running on my computer, while distance sequence actually give me a very large file. So I'll leave that up to you. Okay, so the next one will be the actual analysis of the file. Just now we just basically look at OTU tables, and that's kind of it. The next step is actually just look at what is actually inside and what's the significance of it. So before we do that, we're going to do a subgroup, which is we are trying to reduce our sample so that we don't have such a big file to work with. As you can see that these are the different sample that we have, F3D something something something, and these are the different sequence that we have. We're going to only use the smallest amount. Size of the smallest group is 2580. So what we're going to do is actually subset those sample so that we only have the largest, we're only going to have the largest of 2580, because it's just not it's easier to cross compare them and analyze them, and most of them actually contains a large sequences, but they're mapped very, very low number of times. So we're gonna just throw away all of them and take 2580 and call it a day. Okay, so again, if you're using different sample, the size is gonna change, but here they're using the smallest the smallest size among all the group, so that for the next step of the analysis. Okay, so the first one we're going to do is the alpha diversity analysis and beta diversity analysis. What does that actually means is I'm going to actually borrow this slide from traininggalaxyproject.org, which I believe has a really great explanation of what it means. So you can also find a link to this website down in the video description down below. So concept of alpha diversity is not here. So the concept of alpha diversity and beta diversity is alpha diversity, how many times does a thing appear inside the sample, right? Beta diversity is between the two sample, how different they are. So alpha diversity is usually linked to something called a rarefaction. So a rarefaction is the concept that how many coverage that you have based on your sequencing, based on some complex algorithm that I'm not gonna go into. But usually when you plot out the rarefaction graph, you get these three different shapes. So in the red one, the clones increase while the OTU is continue to increase. Means that if you increase the clone, it's likely that you're gonna get more OTU. So what that means that you might need to get more data, you have to sequence more because there's more data that you will miss if you don't continue to get more clones, for example. So the green one, for example, is the opposite of the red one, where we have so many clones, our OTU does not increase any further. So the coverage is more than enough. So when you do like a flat line that you see on the green line, that's kind of a waste of resources and you should cut back a little bit. So the ideal one is actually the blue line over here, which is straight up, and actually you can see a little bit curved to the flat line there. So that will be the optimal way of sequencing and coverage. Okay, so you can actually see from here, this is an example of the other sample that this project have done. Most of the sample will have a straight up curve, and then that actually flatline off without going further too far away on a flat line. So you can see one flat line over here might be a little bit over sequenced, so we do not need that much depth. Okay, so in this case, Bada has a slightly easier way to analyze alpha diversity by using rarefaction single, and then instead of plotting the graph like we said just now, they're gonna use a summary single function over here that actually is able to tell you the what is that called? Is able to tell you the coverage of each of the sample in a more numerical format. Okay, so what we can do is that we have final opti MCC group AEV STD summary. What we're going to look at is the AVE. Okay, so I also written a simple script right here to actually look in the rarefaction. So let's just have a look. So what we have is a rarefaction coverage. Let's look at right here. So you can see we have 19 individual samples here, as you can see from the group, and the method they use AVE. Again, not going to go into number of sequence 2580, and you can see the coverage of the sequence is 94, 93, 94, 92, and so on and so forth. So even if we have reduced, for example, one sample has 18,000 sequence, we reduce down to 2,500. The coverage is actually good enough. We're not worried about losing a lot of diversity within our sample. So 2580 is good enough and good to go. Okay, let's move on to our next step, which is the beta diversity. Yes, so the beta diversity measurement. So what does it mean by beta diversity measurement? Beta diversity measurement is how different from one sample to another. So they're going to use something very similar. It's actually called a principal component analysis to actually figure out how far away how far away one sequence is from another, and in the end we would want to have something like a PCA plot on the what's that called, the relationship between each of our sample. So let's just actually run the distance share over here and run the PCOA. So once you run the PCOA, you realize that they have PCOA axis and PCOA loading. So let's have a look at what that actually means. So for those that don't understand PCA yet, they're kind of a multi-dimensional reduction tool so that you can plot a 9-dimensional, in this case 19-dimensional plot onto 2-dimensional, and you're able to see how far one sample is away from another. So you can actually see the loading of the axis 1 and 2 is 39 and 80%, so that's 40, 50, about 60%. So a 2-dimensional graph should be able to cover 60% of the separation between the sample, while you have all the axes one by one over here. Again, very difficult to understand, so we're not going to do it here. I also written something in R that can actually read the delimited file that we opened just now and actually plot it in a PCA plot right here. So this will kind of tell you that, yeah, you have 4 distinct cluster where I have one on the left. You can see these are very similar to each other, and all this up here, down here, and down there, they're more or less similar. Remember, the horizontal axis are way more powerful than the vertical axis. So, but they are significant enough that the PCA is able to separate them. So if you have label that you can understand, for example, this is all early 1, 2, 3, late 1, 2, 3, and they're separate properly, then you're good enough. Okay, so if you don't, then you might want to relook into your sample whether they are clustered properly or not. That's kind of a concept of beta diversity in a nutshell. There's a lot more what they call nuance in the analysis down below that I'm not going to go into because they're very, very specific to individual studies. For example, you can get the distance between each of them, which I'm going to show you in the philo tree down below. Okay, so there's also NDMS and all the other thing that we're gonna do. So we're gonna skip over NDMS, but rather we're gonna focus on whether the two groups that we analyze are similar to each other or not. So for that, we're gonna actually assign our group to a kind of treatment because we have not actually done that. So what we're gonna do is that we're gonna create a text file. In this case, the text file should be called design mouse time design. Okay, so what you do is you go to your folder, just create a new what's that called, a text document right here, and then rename the text document into mouse time and design. So let's just look at our mouse time and design right here. Mouse time and design, yeah, right here. So yeah, just copy paste exactly this into the thing where the first row should be a group, the first row should be a group followed by a space, followed by a treatment. And then you actually include all the name of your group accordingly to your batch file that are generated in the first step and give them a different label, either late or early, A or B, or whatever, and so on and so forth. So whatever that you want. So if you don't know what is your group name or you don't know what you have loaded in, you can also find the stability.files, if I'm not wrong. Stability.files here. Okay, so you can actually see from the stability files initially when you load in sample, they will have a list of the name of the group that you have. Just remove the mock and add in the first row and remove all the FASTQ and add in the treatment. Then you should be able to further analyze them. So what this study is actually trying to do is trying to figure out early and late, these two different set of metagenome, these two different batch of bacteria, are they actually different from each other enough or not? So what we're gonna do is that we're gonna run the AMOVA, and we're gonna actually have something like a p-value. So if you look at the p-value here is 0.089. They are not significantly different from each other, means that I might need to look at our sample or assignment, and obviously I think I've assigned them wrongly, which I should be able to reassign them again. Let's see, mouse time design. So yeah, the higher number should correspond to a thing, and the higher number should correspond to late, while the lower number should correspond to early. So let's just actually change it right here and then see if we can actually get any kind of difference between them or not. So these are lower number, so they're early, lower number, lower number, lower number, lower number, lower number, and we have late for 65 and 141. So yeah, it's an unequal amount because I didn't actually consider all of this. So yeah, when I load in the data, I actually base it on the or the size, so that I choose a smaller size of them so that I can get a significant result. But I did not consider that, yeah, it might not actually cause a significant difference. So after I've changed it, the p-value becomes 0.013. So they are more significant, which kind of makes sense. But I believe that if you can follow exactly with the sample that is being hand to you on this MISAC SOP, that you should able to get exactly the p-value that have, which is 0.001 percent. Okay, mine is slightly different because I want my graph to look better, basically. Okay, so that's basically the end of the MISAC SOP I'm gonna follow, and we're gonna actually move on to some other tools. And if you do want to have a more, what is that called, more analysis that follows the MISAC SOP, go ahead and actually just open their website, and then you can again do what I do in the first part one, where you just copy paste each of the thing and then just paste it in your mother. Just make sure that you do not miss a single step, because if you miss a single step, the file name goes wrong, everything will stop running. Okay, so that's basically my SOP, but we are not going to stop here. So the next step, if you base on the thing that we have, we actually done all this, we're going to plot our trees in RStudio. So what you need to do is, again, open the script that I have, and I've included a little bit of script right here for you to actually run within Model. So what you do is you have to run a cluster split based on the FASTA. So let's just run that. Okay, once you've done that, you have to do a make share. And then the most important part is actually do a tree share. Okay, a tree share command is supposed to help you to make a .tre file, and the .tre file is what is gonna be logged. It'll be logged into R later for the plotting. Yes, so cluster split is gonna take a while, so it should be done now. Okay, so that's done. So we have final this and this one and this one. Then we're gonna do a make share based on the thing that we have done just now. And then once we're done, again, we're gonna do a final of the MCC share, which is exactly what we did with our OTU table just now. We're gonna use that and generate our tree share over here. Okay, so these are the two files that we need, the TRE file. Okay, so once you run a TRE file, the TRE file is actually very difficult to understand because they are based on a, I wouldn't say very old format, but kind of old format that is very difficult to understand. It's basically this sample and this sample. What are the distance between them? But the way that they phrase it is not easy to understand for myself, especially. But they're called something called Newick format. Yeah, I should delete this too. They're not really needed in this case. Okay, so I'm gonna use the TAYC file .tre, and I'm gonna load it into R using a package called tree.io. Okay, so once you load in the tree.io, you're going to use a read Newick format to actually include this file. So if you don't know how to install things in Bioconductor, I will just try to include the code down below in the video description, so you can actually include. I'll add a new line over there, so for those that have not installed this thing, you can actually try to install. But you go to Bioconductor website, find tree.io, and there's a script that you can run. So once you've done that, we're gonna include that using the read Newick command, and you're gonna create a list of 5 which have different node and ages. So the nodes are the individual point or the branches in that tree, while the ages are how long that those two is supposed to be. Okay, so that's very similar to like a graph theory situation. And once I've done that, I'm gonna normalize the data. They're not actually necessary, but it makes the graph look slightly better. I'm gonna change the label to a new one, and then we're gonna run ggtree on it. So the concept of ggtree is very simple. You load in your object, and that's kind of it. But however, if you only load in your object, let me just show you right here. If you only load in your object and nothing else, then obviously you get nothing on your graph. But obviously we want to have like labeling, we have to have size and all that. But you can already see this tree sample is very far from the rest on, and so on and so forth. But we don't know which one is which. So we're gonna add something called a geom text. So ggtree is part of it's an extension of ggplot2, so we can actually reuse most of the syntax. So we're gonna run geom text with a new label that I created just now, and we're gonna have this. Okay, so if you zoom in, you can see that all these are all the labeling and how different they are from each other. And you kind of can see that a higher number tends to group together, while the lower number tends to actually group together quite well. So of course, this is not the only thing we want to do. We also want to make sure we can change. For example, this is the ladle size. We actually change the way of how it looks. So just now it is what is that called? It is longer on the bottom, and now it is like longer on top. So it's just a different way of presenting the same tree, but I think more people can actually correct me on that. So the next one, once we're done, is to actually change it to a rectangular to a circular layout, because I believe most of the metagenomics wants it as a circular layout. It's a better looking, more comprehensive look on the different sample that we have and the distance between them. But of course, it's not actually nice enough. We're gonna try to customize it more by adding colors, adding clads, and some annotation and so on. But before we know that, we have to understand which are the branches that we're gonna look into. So I create a sequence called label manual on a sequence to 1 to 37, so we can understand that inside our plot that we have actually export over here, which are the corresponding number to each of the nodes that's inside this graph right here. Okay, so I'm gonna do that, otherwise my phone is gonna close itself. Okay, so the number 1 to 37 correspond individually all the nodes. So if you want to change any information nodes, you can always refer to this file. So what we're gonna do next is actually change we're gonna add something called a clad lab, the labeling for the clad. So we're gonna change it on node 5. So it's gonna be right here. So if you're referring just now, you can actually see that node 5 is right here. So the clad is gonna be here. So if you want it on maybe 27, you can again change this to 27, and you can actually change the direction of the clad very easily on where you want to label it, and you can use the offset to actually change it up and down and do a vertical and horizontal adjust based on that. So do try around with all this thing, and you don't know what else you can change, just press comma, press tab, and you can actually see more mapping and situation that you can actually change within this command right here. Okay, so the last one I'm going to show you is actually the highlight of it, which is here. So I'm going to do it on node 30 and 21 on red and green. Obviously, you will need to choose which one do you want to highlight for your own research. But again, if you don't know the node number, you can again refer to this graph just now. I'm going to do 30 and 21. You can see that 21 is right here, 30 is right here, which is why when I color it, it's going to be up there and down here with a green and a red respectively. So that's basically ggtree, and there's a lot more customization option within this package, which I'm going to make a dedicated video specifically just to explain all that. And there's a lot more you can change, the size, the color of the tree, and there's a lot of different output on that. But for now, we're gonna close on ggtree, and we're gonna move into Galaxy, and we're gonna use Galaxy and Krona to actually further illustrate our data. So so basically, so far what we have is all the tables, and we also can see the relationship between our sample using PCA, the principal component analysis, and we also have done a little bit of ggtree with a phylogenetic relationship. But those are all relationship in between sample. The taxonomy and the information are not very clear. So this is supposed to help them to understand that. So first of all is to actually upload two files, the the final opti MCC share and the final opti MCC 0.03 construct taxonomy. So to upload data into Galaxy, just press upload data and go to choose local file and actually just choose the file that you want. So in this case, you can upload the share file and the taxonomy file, which I'm not gonna able to find right here, but do find the two files that I showed you just now and do a start, and that should upload them into your environment right here. So they should show up green on the right side. So once you've done that, we're going to focus on our taxonomy first to make the Krona chart. So go to tools and then search Krona, Krona. So the search takes a little bit longer than the old version, but it shouldn't be too bad. Yeah, there's a big update if you don't really know for Galaxy, and because Galaxy is not maintained by myself and there's no rollback to the previous version, I cannot ensure that it will work for you, which is why I'm going to include more tools. So the first thing we're going to do is a taxonomy to Krona file. We're going to load in our taxonomy and we don't change anything and we're going to execute. So once this done execute, you have this number 25 over here. So if you have a look, they're actually just converting the taxonomy file into a Krona file, which is very similar, but I think it's just adding columns here and there so that it's easier to convert. As you can see, they're almost exactly the same, right? Compared to the taxonomy here, it's just that they removed the OTU column right in the front. Yeah, right here. Okay, so a slight changes, a slight alteration in the format. So the next one is we're gonna use Krona pie chart, and we're gonna use a tabular, and we're gonna use a Krona formatted taxonomy file, and we're gonna press execute. So once you execute, again, the number 26 here, in my environment is 26, in yours it might be a different number. You will show up something like HTML. Just go to see the HTML. Take note that it might take maybe 1 to 2 minutes for it to run, but it should actually show up in the end, and you should be able to just open it up directly. So again, we can see that we have 15% of lactose we have 6% of this bar C something, and we have this unclassified 50%, and we have so many of different so we can actually have a really good understanding of the population taxonomy within our sample group with Krona, with like really little number of clicks, and we can just upload. And once you've done that, you can just download this file, I think snapshot, and you can download this file and then use it for any of the analysis presentation that you want fairly easily inside Galaxy without any other installation and so on. Okay, so once we've done that, we're going to actually do a make biome file, and we're going to move on to Megan. So if those that don't know, Megan is actually like oh no, this is not the Megan that I am referring to. So yeah, it's actually a bio tools that's designed to visualize taxonomy data. This is also not the tool that I'm referring to. I think it's this one, Compunomics and Megan 6. Okay. Yep, I think this should be the one. If I'm not wrong, they are all free. Yeah, okay. Megan Community Edition is free. You can download, and if you are under institution and you have more money, you can go for the Ultimate Edition and do further analysis on that. But in this case, I'm gonna use Community Edition because it's free, and I believe there's enough things for most people to actually run. So you can download it on GitHub and install on your computer. It runs on Mac, Linux, and PC very easily. Okay, so the next thing we're gonna do after you have installed the Megan is to actually in Galaxy search for make biome. So just to be clear, you can actually run make biome inside Mother, but at the current version that I'm running on, the biome file generated from Mother doesn't work with Megan, which is why I result into using Galaxy to run that. So once we're done, we're gonna use a make biome, and we're gonna choose the OTU share file, and we're gonna use a taxonomy file, and we're not gonna include a metadata in this case because I realized that not needed. So when you're done, go to group and select all the different group, and then press execute, and then it will give you a make biome file right here, and you can actually go to make biome and then just download the file. So a download will give you a biome file, and you just download into your desktop or whatever. And once you've done that, you will be able to go down and have a look at the biome file right here. So a biome file will look somewhat like that. If you don't have all this information in your biome file, you might need to reconsider what you have done, and actually you might have missed out certain steps right here because it might be empty. Okay, or you might need to change your sample input and so on and so forth. So the next step I'm gonna do is to open Megan, which is just another program. So that's very easily. There's some messages, you can have a look, but I'm not gonna do that. And go to open, file, and import biome format. Okay, so I'm gonna choose the biome that I have exported just now in my desktop. Press open, and the data type is gonna be taxonomy. So choose taxonomy, and you can see that I have done a a much more comprehensive phylogenetic tree over here with the different sample as the, what is that called? Of the different color representing the different sample here with more like bacteria, bacilli, and so on and so forth. So again, this is not the only one you can do. You can also change the outlook of your phylogenetic tree. You can also change the way of how you want your sample to be presented. I think my favorite is like this one and so on. I think yeah, it's one of this. I think there's a lot more customization option. You can also go into here and then go to show attribution correlation plot, and you can actually play around with all the different plot that you can see inside Megan over here with the relationship between the different species, the different sample, the composition of bacteria in different sample. In this case, you can also see like that, which is like the abundance of each of the bacteria species. And you can have a sandwich, you can have this one, you can have this one that actually give you all the different, oh yeah, this is so far my favorite. the abundance of each of the bacteria in each sample as its words. And I think you can also do something like change the different color, and I believe that's kind of everything that I have been through. And I think that's everything that I have to show you. And I hope that that's enough to play around. And I definitely have found, again, something new, even just recording this video. Yeah, this is very interesting. Yeah, there's a lot more things that I need to look into. Yeah, domain, rank, let's go for order. And you can actually change the rank to genus so that you get a lot more abundance of all this thing. You can change bubble to have all the look and other stuff. Okay, that's very interesting. I believe you can change the rank right here as well. No, I cannot do that. I can only do it right here. So if I change it to class, what will happen? And then if I do it with bubble, and then I can change to species. Oh yeah, it definitely reduce the total number of things you want to look at, the higher rank that you go. Okay, so that's basically the part 2 of the analysis. I believe we have showed every single thing that I've shown you inside the slide. Just for a cover, we explore the output from mother, the FASTA file, the taxonomy file, and the count file, how to import them into R, how to do basic OTU classification with like distance, sect, and cluster. We done a phylotype on the clustering. We done a phylogenetic on the alpha and beta diversity, particularly alpha with the refraction graph and beta with the PCOA. And we have done plotting trees in RStudio with ggtrees and how to do some simple customization. We have do graphing in Galaxies using Krona, and we use Galaxy to do a make biome and export the biome file to our desktop. And lastly, I use the biome file to do all the visualization I have in Megan. And we're gonna have a look again because they are kind of pretty. And that's kind of everything that I want to say for today. Again, I am not super familiar with the metagenomics analysis, and I'm sure I have missed out certain things that are important, or might have misinterpreted a certain thing. And if you do understand or catch those mistakes, again, leave a comment down below. Or if you have certain analysis that you want to see me do leave a comment down below as well. That certainly helps. Like the video, share the video. Again, it's gonna help a lot with the channel just to keep it alive and so on. And I believe that's all I have to say for today. Again, I want to say thank you so much for watching. It's again another long video, and I hope to see you in the next one. And I'm gonna make sure a lot more videos coming soon. Okay, that's all. Bye.